thank you very much. It's great to be here. I am amazed at how many people whose work I've followed and admired over the years are all at Emory. I somehow hadn't realized what a hotbed this was of fantastic cognitive neuroscience, and now with Paul's arrival, neuroethics as well. So I'm very grateful to be able to present some sort of new thoughts. This is a new talk. It's so new, its age can be measured in minutes. And it actually fits in really well with the ideas that Greg and Stefan were presenting to you. And it has to do with criticisms of brain imaging, fMRI in particular, that we've been hearing more and more about lately. So, you know, you can't open a newspaper or a magazine without seeing brain imaging. When Time wants to, you know, do a feature on the psychology of moral reasoning, what goes on their cover? A false color brain image. And I think because functional imaging has gotten so much attention, and you know, the best graduate students, and lots of funding, there's been a little bit of a backlash where people have criticized either, you know, specific studies, specific practices, fair enough, but in some cases, just these very broad brush blanket critiques of brain imaging. It reminds me a little bit, I don't know how many people here are old enough and watched television in the 80s, but do you remember this shampoo commercial where this model says, "Don't hate me because I'm beautiful"? You know, I think neuroimaging has been so successful that I think it's engendered a certain amount of resentment and people looking for reasons to dismiss it. So before I talk about the critiques, I'm going to very quickly run through the neuroimaging 101 or kindergarten neuroimaging, partly because you know, the critiques tend to focus in part on just what a multi-step process this is and how many inferences are involved. So for any kind of functional neuroimaging, you have some sort of a tracer, you could call it, that's related to neuronal activity. In fMRI, it's the magnetic properties of the blood itself. For bold, you know, differences in susceptibility of oxygenated and deoxygenated hemoglobin. You have a method that can localize that you know, hemoglobin or whatever the tracer is, and reconstruct its position in 3D. You have a bunch of sort of pre-processing, you know, aligning with structural images. There's typically some smoothing done, some filtering. Then you have a whole slew of the kinds of statistics that, you know, cognitive psychologists are more familiar with, testing for activation differences as a function of the task that the person is doing. And with that comes a whole slew of other decisions about thresholds, regions of interest, and other issues that are mainly focused on, you know, avoiding type 1 errors, avoiding thinking you have something when you don't. Probably enough said at this point. Okay. Plus, in addition, lately we've seen a lot of, you know, new techniques, functional connectivity. So you're not just looking at what activates and what doesn't, but you're looking at the extent to which the patterns of activation are correlated between different brain areas. That indicates that those areas are working together. It gives you a more holistic kind of information. We have diffusion tensor imaging, which I guess isn't strictly speaking functional, but uses many of the same instrumentation and gives you, again, pictures of connectivity between areas. And all the exciting work that's been done on pattern classifiers. I couldn't really find a good picture, you know, a nice single image that captures that, but these applications of machine learning techniques, multivariate approaches to image analysis that have been just amazingly successful at approaching the kind of mind reading applications that neuroethics cares a lot about. Okay, so there have been a bunch of reasonable criticisms and concerns raised from within and without the field of functional imaging. There's the famous, dead salmon, who, if you put him in a scanner and fail to correct for multiple comparisons, seems to have brain activity. Okay, this is basically just a sort of colorful reminder that you do need to correct for multiple comparisons. There's tens of thousands of voxels in a typical brain image, and by chance alone, if you're using some conventional significance cutoff like 0.001, by chance alone you're going to have a lot of voxels exceed your threshold, as happened with the salmon. I think it's fair to say that in the early days of fMRI, there were studies published that did not pay sufficient attention, and there probably still are, to these kinds of corrections. But it's not an intrinsic problem with the method, and in general, people handle it appropriately. There are subtler problems involved in the calculation of reliability, statistical significance. Over here, I don't know how many people I'm assuming most of you are neuroscientists, so the famous voodoo correlations paper, which before it was published, the editor insisted that the authors change the title to puzzlingly high correlations, where they basically, skewered a bunch of researchers, mainly in the field of social neuroscience, for statistical approaches that overestimated the significance of their findings or the size of the correlations that they were finding between the psychological states or traits and the brain. This came out, I think, in '09. So this is, fairly recent, but clearly the science is self-correcting here. A similar related point was made by Krieg's court. He used the word double dipping, but clearly there are issues. We have not always been as good statisticians as we should be, but the issues are clear, and most people are doing the right thing, doing it the right way. Another problematic practice for which functional imaging studies have been dinged, and probably deservedly, is what Stefan talked about: reverse inference. What does reverse inference mean? It's a little bit of jargon. Basically, the idea is that forward inference is when you put somebody in a psychological task where you induce a certain mental process, and then you follow the arrow to infer, by looking at your image, what brain areas are involved. Okay, so reverse inference is when you find activation in a certain brain area and say, oh, it must be psychological process 1. And the reason that's problematic is that there's a many-to-one mapping, really a many-to-many mapping between psychological processes and brain areas. But in the case of reverse inference, you would be wrong to infer that it must be mental process 1, having seen brain region A light up, because mental process 2 also can activate brain region A, and maybe in this case it was brain region mental process 2. So to make this a little more concrete, we'll take the study that everybody loves to hate. This is the Iacoboni et al. FKF applied research, political neuromarketing study. Here they found that these uncommitted swing voters, when looking at a picture of John Edwards, showed insula activation. And this was prescient. This was, I think, 2000, when was this? Anyway, before Game Change and the other recent books have come out about John Edwards, may have been, maybe people's insulas knew more than the rest of them. But anyway, they inferred from this that Edwards had a problem, namely, when voters looked at him, they felt disgust. Okay. Well, the problem with that inference is that the insula, very interesting brain area that's involved in all kinds of processes. Disgust is one of them, but there's lots of others. And so it was a huge non sequitur to go from insula activation to disgust. And this is a practice that some people indulge in a lot, reverse inference. We all probably indulge in it a little bit. I think as Stefan said, fair enough when you're sort of doing exploratory work and looking for hypotheses to then test in a more rigorous way. But again, a reasonable criticism that I think the field is responding to. Another thing that we periodically need to be reminded of is that just because a part of the brain becomes active when you're engaged in a certain process doesn't mean that it's necessary for that process. The causal role of brain areas in achieving a certain behavioral outcome or implementing a certain mental process is not made clear by brain imaging alone. So this is a data from a study that I did with Leslie Fellows, where we took patients who had lesions of the anterior cingulate cortex. The color there represents how many of the patients in the group had damage in that given location. And as you can see, with where the red is, everybody had their ACC blown out. In fact, there was one patient who had a huge bilateral anterior cingulate cortex lesion. And so we said, okay, now we know from scores of imaging studies, in fact, I don't know if you can see that there are little tiny white circles here, plotted here. Those little tiny white circles are the maxima, the activation maxima of studies in which the task comparison that the researchers were making, in theory, allowed them to isolate the parts of the brain involved in cognitive control, a sort of certain type of attentional process. We don't need to go into the details on it. But as you can see, there's a lot of data from, pooled from many studies from different labs. One thing you might say is, if we know anything about the anterior cingulate cortex, it's important for cognitive control and cognitive conflict resolution. But clearly, so you would expect people with big holes in their head there would not be able to exert cognitive control and not be able to perform tasks in which there's high cognitive conflict. But what we found, long story short, was no detectable impairment at all. Okay, it raises the question, well then, why is the anterior cingulate cortex lighting up in these, in all these other studies? And the answer to that is, I think, unknown. I certainly don't know the answer. But the point is that clearly necessity, we're not learning anything about the necessity of brain regions just by imaging alone. And finally, I think a very important problem from a neuroethical point of view is just the temptation to take these, very scientific, impressive-looking images and try to commercialize work with fMRI. And this is, I'm picking on them partly because the name of this company always gets a giggle. It's No Lie MRI. But they are one of, a number of companies that are, I think, pretty clearly prematurely marketing services based on fMRI. Okay, this is a lie detection company. There is no evidence that in any sort of real-world context, the method that they use is able to discriminate lies from truth. The data hasn't been gathered. We have data from very simplified laboratory tasks under very artificial conditions. So I think these are all reasonable problems with fMRI. You might call them growing pains, if you will, fair enough. But what I want to turn to are some, I think, not so reasonable critiques. And I'm just, rather than try to put up a lot of text up there and then read it aloud and have you reading at a different pace and be very aggravating, I'm going to just read you some excerpts from just these different sources. So the first is from the journal Bioethics, article by Christian Huber and Johann Huber, called Epistemological Considerations on Neuroimaging, a Crucial Prerequisite for Neuroethics. These people are pretty measured, but they basically say a high degree of objectivity is attributed to neuroimages. But on the contrary, figures from neuroimaging papers provide visualizations generated from indirect measurement of surrogate parameters from of mental processes. They are produced in highly differentiated processes involving probabilities and subjective choices, are influenced by the hypotheses raised and the questions asked at the beginning of the experiment, and are interpreted in this context. Okay, fair enough. It doesn't seem like that's beating up unfairly, although you might say, well, you could pretty much say the same thing about any scientific measurement. Radio astronomy is whatever. Okay. Here's something from Scientific American. It's an article by Michael Shermer, who sort of specializes in giving the skeptic's view of various things. It's entitled 5 Ways That Brain Scans Mislead Us. Subtitle, Colorful Scans Have Lulled Us into an Oversimplified Conception of the Brain. He says, here's 5 flaws in brain scans. Okay, the first one is they study cognition in an unnatural environment. Okay, fair enough. Scans are indirect measurements of brain activity. One often reads popular accounts of fMRI research describing how the brain lights up when thinking about money or sex or God or whatever. But here's what's really going on. And then he talks about the bold signal and how it's not neuronal activity at all, it's plumbing and so forth. Next, colors exaggerate the effects in the brain. Pictures of brains splotched with sharply defined color regions are highly misleading because they suggest well-defined processing blocks. The coloring is artificial. I mean, would anybody really think that it's not? I don't know. And the process of coloring the regions is even more misleading. As Churchland says, and they got Pat Churchland to say some things here, which I suspect were out of context because I don't think she really... the difference in activity levels is tiny. You can make these differences look huge by coloring them red and by subtracting everything else out, so it gives an impression that is exaggerated. The choice of what to emphasize is also misleading. Take the cingulate cortex, an area dealing with conflict. You can get it to, okay. And then she just goes to talk about, again, that study that we all love to hate. Okay. Brain images are statistical compilations. And they go on by saying, you're not looking at any particular person. Well, in fact, sometimes you are, but You're not looking at any particular neural event. It's all averages and trends and so forth. Again, I'm not sure if that's a plus or a minus. Might be a plus. And then he says, keep all this background in mind the next time you see one of those colorful brain scans with an arrow pointing to some spot that says, this is your brain on X. The image usually does not represent any one person's brain. It is a statistical computation of the entire subject pool rendered with artificial colors to highlight the places, blah, 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 blah. Okay. Okay, here's a piece published in The Scientist, and it's called Fake Method for Research Impartiality, fMRI. Behavioral science's bid for enhanced status falls short. There's one problem with this new wave of brain science. It's still not really science. The reading of fMRI scans is still a highly interpretive practice. So again, this theme of it's subjective, it's the context, it's just kind of what the researcher just reads out what he wants to. Sometimes the difference between seeing higher activity in the parietal lobe compared to the occipital lobe is akin to deciding whether Van Gogh or Matisse is the more colorful artist. And then she goes on to say a far greater limitation of fMRI is that it measures blood flow, whereas it might take milliseconds to decide to place a blah, 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 blah. It takes much longer for the blood to flow, et cetera. Okay. The New Atlantis. This author, Matthew Crawford, starts by, he talks about this kind of research as a dramatic imitation of explanatory competence that wows us and cows us with its self-confidence. He talking about the fact that the bold signal is not actually the firing of neurons. He says, from this point of view, the signs of brain activity obtained from the scanning system are no more codes of what is going on than any other physiological correlate, such as the electrodermal response or an electromyogram. Let's see, there's one other point that he makes in here. Oh, perhaps the most fundamental limitation of functional imaging vis-a-vis the claim that it allows us to peer inside the mind is that there is a basic disconnect of time scale. Brain scans are emphatically not images of cognition and process, as the neural activity of interest occurs on a time scale orders of magnitude faster than the hemodynamic response, the proxy for neural activity measured by MRI. Okay, courage. There's only one more thing I want to read to you. And this is from Jerry Fodor, who you may know is a very influential philosopher of mind. And he's writing in the London Review of Books this little sort of grape piece about how he loves to read the New York Times science section every Tuesday. He's an addict, but it really bugs him to see how much coverage they are giving to fMRI. It's functional localization by neuroimaging, for which the Times is especially enthusiastic. And I guess that as the times go, so go the grants. It particularly likes those polychrome maps that show a place in the brain that's red when you're thinking about one thing and green when you're thinking about something else. I wonder why the Times cares. I wonder why anyone cares. I'm not old enough to be surprised when people are interested in things that seem to me not to be interesting. Golf, the music of Andrew Lloyd Webber, and the adulteries of politicians are three examples in a plethora. Since in most cases we're not competing for resources, I don't mind at all. But science is different. Science is expensive, and it's largely publicly funded, and there's never enough money to do all the research that might be worth doing. In particular, brain imaging is expensive compared to other ways of trying to find out about the mind. If you put your money, which is to say our money, into the elaborate technology required to establish neural localizations of mental functions by imaging techniques, you almost certainly take it out of other kinds of psychological research. So, fMRI is really getting pummeled here on various grounds. And then Stephen Rose, who you may know, he's a neuroscientist from England, works on memory and the brain. But he's a little bit of a, what's the word, curmudgeon. He basically does animal research, and he's a little bit of a curmudgeon about human neuroscience and imaging. And he says Jerry Fodor's provocative dismissal of brain imaging scores some palpable hits, but misses the main target. Yes, the false color images are marvelously seductive, but by the time you see them, they have been so massaged as to risk being thoroughly misleading. Okay, so let me just sort of sum up, the themes here. First of all, when imagers are getting all the money and resources, these images aren't really images of brain activity; they're images of blood flow. There's a lot of statistics going on. In fact, there's kind of massaging going on. There are subjective decisions going into how the data are processed and what they ultimately show. Those are probably the main things. So let me just go through each of those and try to respond to them. First of all, the idea that it's not really the brain that we're looking at, it's the blood in the brain. Think of any science you want, and the measures, the signals that are being recorded, the dependent measures are not the things, are not the subjects of study, right? This just seems like an odd criticism a priori, right? Those silly chemists thinking that they're learning something about the chemical composition of something when really they're just measuring absorption spectra, right? You can make this criticism about spectroscopy, about astronomy, about anything. But furthermore, it's an empirical question how well the bold signal is related to brain activity. And it's an empirical question that we know a lot about. And it's, depending on, it relates to some aspects of brain activity and not so much to others. One person, I think, who's known for being kind of the most critical but also constructive in terms of finding out what the signal is measuring is Logothetis. This is an image from one of his papers, basically looking at different aspects of neural behavior and what is most strongly reflected in the bold response, and LFP, which I believe is localized field potential. Is that right? I'm a little out of my depth here in terms of the actual physiology, but the point is there are kind of known measurable properties of brain activity that are reflected in the bold signal. And as long as we know that and have ways of checking the correspondence, this idea that these silly people, they're mistaking blood flow for neural activity seems a very misplaced criticism. Another criticism is, you're actually looking at maps of statistical results as opposed to just a simple direct measurement, or it's all a construction. These are highly processed images. Well, presumably you want statistics, right? That seems odd to sort of feel like you're getting something less reliable, less certain by having maps that have exceeded, that have been subjected to statistical analysis as opposed to just a representative measure or something. But in addition, neuroimaging is not the only field where there are legitimate issues to be raised and to be dealt with concerning how much manipulation for what purposes you should do in an image. And maybe I'll spare you more reading aloud, but basically, I have found in a couple of cell biology journals very interesting discussions of what things you can and can't do with Photoshop with your images prior to publication. Okay, basically the guidelines are that if it's making the kind of the hypothesis that if it's making the relevant evidence clearer, easier to understand, and you state what you have done, there's nothing wrong with it, which makes good sense. Okay. Finally, let me talk about a criticism that has gotten a lot of airtime lately. It's discussed a lot in the realm of neuroscience and law because people worry about juries and judges being overly influenced by brain imaging. But this idea that you see this really impressive colored image and you kind of totally lose all ability to be a critical thinker and evaluate the evidence, and you just kind of go, oh yes, I've seen the image, it must be true. There are a few studies, like literally 3, that get cited over and over as evidence for this. And I think one of our earlier speakers, I can't remember, it was Greg or Stefan, said something like, and we know that these images, really strongly sway people. But that's an empirical question, and it turns out we don't have much evidence for it. But let me take you through the evidence that we do have. The first one is Dina Weisberg's paper, which I don't think you can really read this. Well, okay, she gave subjects 4 kinds of scientific explanations for psychological phenomena. And these explanations varied in a 2-by-2 manner. She had good explanations where the psychological effect was actually explained, as a result of other things, other sort of more basic, more fundamental things causing this effect. And she had bad explanations that sort of just, used a bunch of terminology, but didn't really, they were circular. They didn't really explain why the effect obtained. So those are the 2 columns, good explanations and bad explanations. And then she orthogonally varied whether the explanations included any mention of neuroscience or not. Okay, and maybe I should just, if you actually can read that, I'll just, have you guys finished reading it, or? Yeah, okay, good. So the question is, what does the mere presence of a mention of neuroscience, in this case brain imaging, do to people's ratings of how good an explanation it is? Okay, and the hypothesis being tested is that the mere presence of neuroscience, and in this case a mention of a brain scan, will make people think a bad explanation is a good explanation. Okay, it'll make them think they've learned something, they've understood something, just because there's a reference to a brain scan. And that is indeed what she found. But, in cognitive science and cognitive neuroscience, just a little aside, most of our research is based on hypothesis testing and significance testing. Okay, we set up some kind of a experiment where if it comes out one way, this model is true, and if it comes out another way, that model is true. Okay, And then we analyze our results to see if it's significantly this way or significantly that way, because we want to rule in or rule out a model. But sometimes when you're talking about real world situations, what matters is not just is something statistically significant. It also matters how big the effect is, right? If it's a tiny effect, if it's a minuscule effect, even if it's highly significant, it doesn't really matter. It's statistically significant, but it's not socially or in reality significant, okay? And I think that's what we have here, because what this scale doesn't show is that people were rating how good the explanation was on a 7-point scale. And basically, what you see here is that the difference, well, the presence of neuroscience, first of all, what's interesting is it didn't really affect people's ratings of how good the true explanations were, the good explanations were. It only influenced their ratings of the bad explanations, and that's interesting. But it, less than 1 point on a 7-point scale. So I think a fair summary of this study is, yes, there is evidence here that by citing brain imaging evidence, people are more likely to find a bad explanation acceptable or good, but just by a little tiny bit, okay? Less than 1 point on a 7-point scale. I mean, I think that in terms of the critique that, my God, you show somebody a brain image and their critical faculties go out the window and they'll just accept anything, that story is not supported by this data. Okay. Next study, another recent study, McCabe and Castell, published in the journal Cognition. They gave people, again, a little piece of bad science. In this case, a reverse inference. The story is that neuroscientists have shown that watching TV activates certain brain regions, and in fact, it activates the same brain regions in the temporal lobe as doing arithmetic. Therefore, and the question is, is this a good inference? Therefore, watching TV should improve your arithmetic ability. Okay, just patently silly, okay. And then they asked people, do you believe this? How persuasive is this? For some people, that little silly story was accompanied by a bar graph showing temporal lobe activation while watching TV or doing arithmetic. Showing, very similar amounts of activation in the temporal lobe for TV watching and for doing arithmetic. In the brain imaging condition, what they showed is, here's the brain image for watching TV, here's the brain image for doing arithmetic. Both show activation in the temporal lobe, therefore watch TV, it'll improve your arithmetic ability, okay? And what they found, as I'm sure you can guess, is that people who didn't see any illustration at all and people who saw the bar graph were rated the persuasiveness of that argument about the same. But people who saw a brain image actually found it more persuasive. But those two depictions, the bar graph and the brain image, were not equally informative. I mean, frankly, this is silly, right? There's a certain amount of temporal lobe activity. The temporal lobe's a big place in these two conditions, so you want to somehow relate them to each other. That seems preposterous. But you know what? I would spend a couple of minutes thinking about the plausibility of whether TV watching could improve arithmetic, given those data, right? I mean, this is a highly similar pattern of activation across these two cases, right? I mean, I think any sensible person would say, TV watching and arithmetic, that sounds pretty goofy, but boy, there is something in these two conditions that's drawing on very similar neural resources that bears thinking about. So these, the idea of this condition, the bar chart condition, was that it was supposed to be equivalent to the brain image in conveying the same information and conveying it visually. But of course it doesn't convey the same information.